just receive.
Okay, good morning, Gimnasio Moderno. Students, teachers, staffs, and invitees that are watching this morning live through Facebook Live or YouTube Live, or obviously the Gimnasio Moderno website itself. Uh, Today, it is a great honor and pleasure for me to invite you all to a speech about the world's current situation, a lecture given by one of the most prestigious persons and investigators in this area at the moment. So it is a great honor for me to introduce Professor Akiko Iwasaki from Yale School of Medicine. She is a professor of immunobiology and molecular, cellular, and developmental biology from the Waldemar von Zettwitz. She is also a professor and investigator at the Howard Hughes Medical Institute. She has been awarded multiple times for her investigations regarding innate immunity and how mucosal surfaces recognize viruses, involving her studies with viruses as Zika, influenza, or in our case, COVID-19, which are respiratory affections after all. So I could spend the morning telling you about her and her outstanding contributions to science, but it is time for me to step aside and let her with you. So with no further ado, give Dr. Iwasaki a warm welcome. Good morning, Dr. Iwasaki, and welcome to a glimpse of Colombia and the Gimnasio Moderno.
Thank you. Thank you very much, Diego, for your very kind introduction. I am delighted to be here. Good morning, everyone. I believe you're watching me from live stream as well in your homes, and I really welcome you to this session. So it's my great pleasure to talk. Yes, it's my great pleasure to talk about the immune system. And I think I'm going to give a short introduction to the immune system because I think it is very important to understand how our body functions to recognize and fight infections, especially in this pandemic time. So I'm going to share my slides with you. And let's see.
Sure thing.
Okay, you see my slides? Yes? Diego, can you see?
Yes,
Okay,
they are streaming, no worries.
Okay, wonderful. Okay, so
yes, yeah, no worries.
Okay, I'm very delighted to talk about the immune system today. And for those who have access to Twitter, I have almost on a daily basis, I talk about different studies that are coming out in COVID-19 disease and immune response, vaccines, and what antibody levels mean, and various different things. So please follow me in Twitter, Viruses Immunity. So first I want to talk about the immune system and how it recognizes viruses and what it does to counter this infection. So normally, and these discoveries, some of these discoveries were made here at Yale, and this is not a very, very old discovery. It's within the last 20 years or so, we really finally start to understand these mechanisms. So when a viral pathogen enters the host, it's detected by sensors that are either on the surface of the cell or inside of the cell that detects unique features of viruses. And these, much of this feature is actually the nucleic acids that are unique to virus, either DNA or RNA. And those sensors, once they're engaged, signal to the cell into the nucleus and tell the cells to make different proteins. And these different proteins include cytokines and interferons. So this is the first alarm signal that your nose or your lung cells will make in the very beginning of the infection. And these interferons, once
Yes.
they're secreted, it can bind to the receptors on pretty much every cell type of the body, can detect these interferons, because pretty much every cell type of the body can become targets of virus infection. So what it does, it does many different things, this interferon. It's very important. First, it signals the cells to put up defense system so that it can put up barriers for protecting against further infection. It can also signals the cells to die, and infected cells to die. And that's very important because you want to get rid of the source of virus that is creating more virus in the body. And third, what it does is it allows recruitment of cells from the bloodstream, because the infected cells themselves are often not capable of blocking the virus on their own, because viruses actually have multiple mechanisms to block that kind of defense. So what we need is these professional soldiers coming from the blood, swarming the sites of infection to block further spread. And these professional leukocytes, or white blood cells, actually have arsenal of different mechanisms to eat up or block further replication of these viruses. Sorry.
Okay.
Okay. For instance,
That's okay.
it's okay. All right. I just want to talk
Yeah,
about.
it's okay. It's fine. It's fine. Beautiful.
Cytokine storm. So what I told you is that the first step that happens in infected person is the cells that are infected themselves secrete some cytokines, interferons, and that can recruit macrophages or other cell types to come from the blood to fight the infection. And in this case, these macrophages are detecting the elements of the virus that I first talked about in the first slide, and secreting further different cytokines. Now, this is usually secreted to block further spread of the virus. But in the case of SARS-CoV-2 or COVID-19, what happens is that there is this sort of uncontrolled level of cytokine that happen within the lung, and that further recruits more leukocytes from the blood into the lung. And they're all capable of secreting different sets of cytokines and so on. Also, what happens is you have this cytokine storm where many, many more cytokines from many other cell types are secreted. And that causes certain destructions of the tissue, such as the airway cells that are lining the alveolus here are damaged, and fibrin deposit accumulates, as well as leaking of the fluid from the blood into the lung happens. This is really what happens during acute respiratory distress. And when this isn't controlled, a person can die of this type of diseases after the cytokine storm. So cytokines are intended to protect the host, but in this case, because it's too much and too many different cytokines secreted by different cells, it actually results in damage of the tissue and therefore mortality in some cases. So how does the immune system fight this infection if all of these things are happening? Well, there's this other arm of the immunity called the adaptive immunity. And this is really important. This is what gives us the ability to remember different types of infection and to become protected the second time we encounter the same pathogen. So for instance, if you take the virus that's coming in through the lung, you have these lung epithelial cells that are infected. One of the cells that are recruited from the bloodstream that I mentioned, as well as those that are resident within that tissue, are these dendritic cells. And dendritic cells are really important because they take the virus particle or debris from the virus particle, and they can become activated as a result of this activation, innate sensors that I talked about. And once they're activated, they can migrate to the lymph node, which are placed all over our body to survey for infectious pathogens. And within that lymph node, the lymph node are like kidney bean type of structure that you find really throughout the body. But what it is to really screen for different types of antigens and pathogens that are coming into our body and stimulating these lymphocytes. Lymphocytes are specialized white blood cells known as T and B cells, and they have receptors on the surface that detect unique features of virus. And these cells are being stimulated by the dendritic cells that are migrating from the lung into the lymph node. And once they found the match between the T cells and the dendritic cells, these T cells become activated. When B cells detect through their B cell receptor antigen, they also become activated. And they divide and they become effector cells. And once they become effector cells, they exit the lymph node into circulation again. And now they can go back to the tissue, such as the lung, and start attacking the infected cells, as well as block further spread of the virus by secreting antibodies. So this is the sort of adaptive immune system that provides specific immune response against a given pathogen, in this case, this particular virus. So what these antigen presenting cells or dendritic cells, what it does is to educate the lymphocytes to become either killer cells, which are the T cells that detect specific antigen on the surface of infected cells and attack them. And then they also help educate B cells to develop neutralizing antibodies that bind the surface of the virus and block it from coming in contact with the host cell by blocking the engagement of the viral particle and the cell surface receptor. So this is really the basis of vaccines. The whole point of vaccine is to make a very good set of killer cells or antibodies that can protect you from becoming infected or becoming sick from infection. But not only that, as I mentioned, if you're exposed for the first time with a new virus, you develop a certain level of antibody, but that antibody usually wanes, and that's what we see with coronaviruses, is that within about a year, there's a decline of antibody. But that's not too much to worry about, because once you have a second exposure, normally what happens is that you get a much better secondary immune response, where you have a much better concentration of antibody, as well as much more specific and potent antibody response, that the next time you're infected, the third time around, you're probably not going to feel anything, because your antibodies will then protect you from infection altogether. Anyway, so there's this sort of, usually we also use the same concept for vaccination, that usually you have a initial vaccine followed by a booster vaccine. And that's what gives you a very high titer of antibody that can protect you from infection. And just to let you know that, as I mentioned, each lymphocyte has a specific receptor that can detect different pathogens. So a B cell might recognize a certain virus, but each B cell have antibody to different types of viruses. And so what you want to do is to educate the immune system to secrete antibodies that are selective against a particular pathogen. And that's what vaccines do. So a measles vaccine might generate anti-measles antibody. SARS-CoV-2 vaccine will generate anti-SARS-CoV-2 vaccine, and so on and so forth. And that's why we need vaccine against each specific pathogen. And just to reiterate this point, that if you are exposed to the virus once, you might, so this is the perfect example that I was talking about before. The infection will result in generation of lots of T cells and B cells that become memory cells. And so the second time you're infected with the same pathogen, you might have a little bit of replication of the virus, but mostly will become asymptomatic, and that you can really recover from disease without having any symptoms at all. Whereas a completely naive individual, because there's no memory response, the virus will replicate, and you might become symptomatic if that happens. So, there's a lot of talk about antibodies declining against this virus and so on. But the other thing that I want to point out is that a vaccine can give you a much better immune response than a natural infection. And so once we have a very good vaccine, hopefully everyone will be protected in this manner. So finally, I want to end by why do science? So I've been a scientist for a long time, and I love it. This is like the best thing that I can do for my life. First of all, it's really fun to do science. You discover new things every day on different levels.
Indeed.
And it's really fun to discover how nature works. And then second reason to do science is to provide answer to key questions in life. I mean, everyone can, have different curiosities. My curiosity is about viruses and immunity, but you might be interested in how the brain works, or how the heart works, or how the ecology works, or whatever it is. Science provides answers to key questions in life. And finally, it not only is fun and answers to key questions, but it also can help other people. And this is what's really kind of great about science, is that not only do you get to have fun, but you also get to help people. And that's what I feel very privileged right now, to be able to do studies on COVID patients, so that hopefully we can help people recover from this infection by understanding how the immune system works against this virus and developing a better therapy based on that understanding. So I'm going to end here, and I'll take any questions.
Well, that was amazing, and thank you for the brief speech about the immunity. So for making like an abstract of this, and it requires for us to let the students know that we have two kinds of immunity, primarily, right? So we have the innate immunity and the acquired immunity, and those immunities, one of those, we have to train it with vaccines. Is like more or less the way we should do, right?
Correct.
So what can you tell to the students about the vaccine, the promising ideas of the vaccine that we are going to have soon?
Right.
What can you tell the students?
Yeah. So as you may know, there are over 100 different vaccines. I think it's 180 something vaccines right now being tested in various stages of trial. And many of these vaccines are targeting the spike protein of the virus, which is really that protein that I showed you that binds to the surface of the cell to infect the cells. And so this is a very important target of training the adaptive immune response, B cell response and T cell response. And what it does is, when the vaccine is given to a person, as I mentioned, you can develop very good B cell antibody and T cell immunity. And so right now, there are so many different types of vaccines that are being tried. The traditional vaccine, which includes sort of inactivated SARS-CoV-2 virus, as well as mRNA vaccine that you've probably heard about, which really takes the spike protein mRNA and introduces it into your own cells to make the antigen. And also DNA vaccines, very similar concept. Instead of taking the RNA, you take the DNA, put it into the nucleus of the cells, and then allow that cell to make its proteins, the spike proteins. And then there are also the kind of chimeric virus.
It is important. I mean, I believe I believe for me or the students to understand that there are no just one kind of vaccine entirely. I mean, there's no just a vaccine that manages any of the diseases. I mean, it's like the recombinant, maybe the attenuated, the parts of the virus that we use sometimes, and so forth, right?
That's right.
So,
there are multiple
yeah, go ahead. No, I'm sorry.
Yeah, so there are multiple types of vaccines, and each of them has its strengths and weaknesses. But my hope is that after the phase 3 trial, we will know how safe and effective these vaccines are. And the great thing about having more than one vaccine in the pipeline is that, you know, we can make enough of these vaccines to cover the entire people on this earth. Hopefully, if there's only one vaccine, it will be very difficult to do as well. So multiple approaches are needed across the world right now to make enough vaccines for everyone.
Okay, so we have a question from one of the students called Gregorio. As research in genetics advances, it becomes apparent that genetic engineering tools like CRISPR-Cas9 will be indispensable in the fight against diseases. So how do you see the CRISPR-Cas9 playing a role here in the fighting of the virus and the virus to come?
Right. So, I mean, so every technology that's being developed is being applied right now throughout the world, including CRISPR-Cas9 system, as well as other Cas systems. So, for instance, the detection of viral RNA might use some Cas system to detect certain specific sequences of the virus, and that might be even faster than doing a traditional PCR. And obviously, if you need to engineer different cells to express different things or mutate certain things, CRISPR-Cas9 technology is a powerful technology to be able to do that. So the beauty of this is that we are in 2020, and we have a lot more tools than ever before to tackle not only understanding of the immune response, but development of diagnosis and vaccines and therapies. So everything is being used right now.
Sure. There's a question from one of the person that's watching us, and she's asking that she didn't really understand the role of the interferon with the virus. That if you can please clarify a little bit of the interferons.
Sure. So interferons are really key antiviral cytokines. There are multiple interferon families, type 1, type 2, and type 3 interferons. And essentially what it does, as the name suggests, it interferes with the process of virus production and spread. So interferon, when it's released from this cell, can bind to receptor on the neighboring cells. And it induces signals through those interferon receptors to induce hundreds or thousands of genes known as interferon stimulated genes. And many of these genes have direct antiviral activity that can block the replication of the virus RNA, or it can block the assembly of the virus, or it can block the egress of the virus from the cell to infect other cells. And so hopefully with all these different interferon stimulated genes all together can control the spread of the virus in the interferon stimulated cells. And it's a really potent antiviral. And if you have enough interferon early on, you can really control the virus well.
Yeah, so it's like blockers of the linkage for the virus and the host cells. So it's very important. There's another important question here, and I believe it's a very nice question. Camilo Pardo asked, can we pass immunity to any of the diseases and eventually COVID-19 through genes? And if so, how it work? What do you say?
Immunity
I believe it's a very nice and a pretty question. Yeah, through genes, like your offspring.
I see. Like transmit the immune response to your children?
Yeah,
Ah,
more or less is the question. It can
oh, I see.
encompass immunity to any of the disease,
Oh,
and
okay.
eventually COVID-19 through genes, like if the genes are going to get any so adapted or whatever
I
to,
see. Yeah. Well, that's
I
not
don't
how
know,
the
to
immune
pass it.
system works, actually. So, even if I'm immune to COVID, I can't pass that on to my future children because, every person who's born has a completely different repertoire of adaptive immune cells, the lymphocytes.
Uh-huh.
They are completely
All
uneducated.
right.
Yeah, so each
Yeah.
person has to be educated, just like what you learn from classroom. You can't really transmit that to your children. It has to be every person educated through vaccines.
Okay. So again, we are going to be so keen with that part that you have innate immunity and other that you have to train, and part of it is when you are a newborn, and so and you're growing and learning from the different environmental attacks,
Mhm.
right? So you're going to learn from that. Well, there's another question here, and it's Gregorio once again, which is very attentive to this. It's very nice. Do you think that vaccines will become obsolete due to this fact? I mean, it's continuing the question. Or do you think the potential moral risks that come with genetic engineering will prevent it from becoming a common medical practice? So it's a
Okay.
interesting question, right?
Well, I don't think vaccines will become obsolete because we will have pathogens that are emerging and that can cause,
Mm-hmm.
epidemics and pandemics throughout human history. We've dealt with so many different pathogens. So every time there's a pathogen, we have to make a new vaccine, and I don't think that's going to
Mm-hmm.
become obsolete. And also, as I mentioned,
No, no.
every person has to become educated through the vaccine. So it's not that you can inject one miracle drug and become immune to everything.
Yeah, well, good point. So there's no, there's not going to be obsolescence for the vaccines.
Mhm,
Not in the future. We need to create a new vaccine for each one of
mhm.
the illness. So there's another question here, and it says David Diaz asked if the more we get exposed to a virus, the better our response is, then why we are still getting affected by the flu virus, for example? Should not we
Very,
get already be immune to it at this point? And it is an incredible question. I mean, it's a beautiful question
yes. Thank you for
indeed.
that question. So what I didn't talk about is that every virus has its a different strategy to evade immune response. So in the case of flu, what happens is that there is a genetic mutation that occurs every year that allows the virus to escape the antibody from the year before. So this is essentially why we need annual vaccines for flu, because the flu changes every year to escape from antibody detection. And so this is a unique virus, not like coronaviruses. They don't mutate every year like this. Flu is unique in its ability to mutate away from recognition by the antibody. And that's why we need new antigen vaccines every
New
year.
antigen.
Mhm.
That's beautiful. So I have the last questions due to the timing, and it's what's the difference between a reinfection and reactivation of the virus in the same person? Because we are talking about that. I mean, it's a top topic because we are talking about,
Yeah.
are we going to get reinfected by the COVID-19? And if so, how is it going to happen? And so on and so on. So it is very complicated nowadays. And that's a tough topic about the difference between reinfection and reactivation of virus, isn't it?
Yeah. So there's one way to distinguish reinfection and reactivation, which is to sequence the genome of the virus from the first and the second infection. If they're identical, they're or very, very similar. They're probably likely reactivation of the same virus. If there's a reservoir of the virus somewhere that is being reactivated in somehow, we could have such a situation in which there is a resurgence of the same virus. But if the sequence from the first and the second virus genome is very distinct, you know that it's a reinfection, because the second virus, has to have come from another source other than your own self to have become infected. And there are now dozen cases or so of confirmed reinfections that are coming out. And I believe more of these reports will come out as we go further into the pandemic, because as I mentioned, the coronaviruses usually don't give you a lifelong immunity, that the immunity wanes within, a year or so. So it's not surprising to get reinfection. But what it is important to note is whether the reinfection is going to protect you from disease or not. And that's also, right now, the outcomes are very different between each reinfection cases.
Brutal. That's amazing information, Dr. Iwasaki. I'm sorry we have to finish due to timing.
Okay.
And it was an enormous pleasure for me and enormous pleasure for the school, the entire school, to have you here this morning. And we have some other questions there that maybe we're going to write at you for your answer. And thank you so much, Dr. Akiko. That was amazing this morning. Thank you so much.
Thank you very much, Diego. And
thank you so
to all the
much.
students. Very nice to see you.
No, it was amazing. And the students, thank you so much for the questions. They were magnificent questions. Thank you so much for being there. And well, see you next time with
Bye-bye.
Dr. Akiko Iwasaki. Keep an eye on her Twitter. Keep an eye on her Twitter. She's constantly posting information that is very important every day. So keep an eye on her Twitter account. Okay.
Thank you.
So Dr. Akiko, thank you so much.
Okay. Thank you. Have a wonderful day. Bye.
Bye.